হ্যালো স্টুডেন্ট, আজকে ক্লাসটা 6th সেমিস্টার মেজরের টপিকটা তো গতদিন যা করিয়েছিলাম লেক অপারন, আর আজকে জিন রেগুলেশন স্পেশালি লেক অপারনের সঙ্গে রিলেটেড প্রয়েবল কোয়েশ্চন আর সাথে কিছু আনসারও লিখে দিচ্ছি। তোমরা তৈরি থাকো ঝটপট করে। আচ্ছা, প্রথমে ফার্স্ট ইউ রাইট সাম প্রয়েবল কোয়েশ্চনস অন জিন রেগুলেশন অ্যান্ড স্পেশালি লেক অপারন. Write down the questions first. What is central dogma? What is central dogma? What is gene regulation? What is Operon model? What are the main components of Operon model? What are structural genes? What is operator gene? What is promoter gene? What is regulator gene? What is the function of repressor? What is the function of repressor? What is co-repressor? What is co-repressor? What is inducer? What is positive regulation? What is negative regulation? What do you mean by inducible and repressible system? What do you mean by inducible and repressible system? How does repressor act? How does repressor act? Differentiate between inducible and repressible system. Difference between inducible and repressible system. What is operon? Describe Lac Operon with a suitable diagram. I said these are the probable questions related to gene regulation, especially the Lac Operon. তোমরা এর আগের দিন তোমাদেরকে বুঝিয়ে দিয়েছি, ম্যাটেরিয়ালস থেকে সাপ্লাই করা হয়েছে, নিজেরা কিছু করো, আমি কিছু করেও দিচ্ছি। ওকে, নাও ফার্স্ট রাইট হোয়াট ইজ সেন্ট্রাল ডগমা. The flow of information is one way from DNA to RNA to protein. The flow of information is one way from DNA to RNA to protein. This sequence of information transfer between DNA and protein is called central dogma. এটা আমরা কি করে করি? DNA right hand arrow, messenger RNA right hand arrow, protein. DNA to messenger RNA, it is transcription. Messenger RNA to protein, translation. ঠিক আছে? হোয়াট ইজ জিন রেগুলেশন? এটা তোমাদেরকে লেকচারে খুব ভালো করেই বলা হয়েছিল। Now write down the answer about for the question, what is Operon model? A group of closely linked genes which act together and code for various enzymes of a particular biochemical pathway is called operon. The operon model is also known as operon hypothesis. Now, the main components of operon model. The main components of operon model are structural genes, operator gene, promoter gene, regulator or inducer gene, repressor, co-repressor, and inducer. Now let us write down the structural genes. The genes Z, Y, and A in Lac Operon of E. coli which control the synthesis of protein from messenger RNA are called structural genes. Now, let us write down about the operator gene. A gene which is located between promoter and structural genes in operon model of E. coli is called promoter gene. It controls the function of structural genes. Now, let us write on promoter genes. In operon model of E. coli, a gene which is located between regulator gene and operator gene is called promoter gene. The main function of promoter gene is to initiate transcription. Now, write on regulator gene. In operon model of E. coli, a gene which is located on one end of operon near promoter, which is located on one end of operon near promoter gene is called regulator gene. The function of regulator gene is to direct the synthesis of a repressor which is a protein molecule. Now, write on the function of repressor. The function of repressor molecule. The repressor may be either active or inactive. When it is active, it binds with operator gene and prevents transcription and protein synthesis by inactivating promoter gene. When the repressor molecule is inactive, transcription and protein synthesis take place. Now, write the answer of co-repressor. Co-repressor is perhaps a product of one of the enzymes synthesized by structural genes. Co-repressor can active the inactive repressor and prevent transcription and protein synthesis. Now, what is inducer? What is inducer? Inducer is a substrate that is lactose in Lac Operon. Which promotes transcription. It binds with repressor molecule and inactivates the same. The repressor then cannot bind with operator gene. Hence, the transcription and protein synthesis take place. Now, let us know about the positive regulation and negative regulation. Positive regulation. In operant model of E. coli, inhibition of transcription by repressor by inactivation of promoter is called positive regulation. Now write down the negative regulation. In operant model of E. coli, enhancement of transcription by an effector molecule by activation of promoter is called negative regulation. Now, write down the differences between inducible and repressible system. You make two columns. On your left hand right it inducible system, and on the right hand irrepressible system. Left hand inducible system, right hand irrepressible system. Point number 1. Under inducible system, regulator gene can produce active repressible molecule called repressor. Number 1, right hand. Regulator gene produces inactive repressor. Regulator gene produces inactive repressor molecule called aporepressor. Point number 2, I'll say only left hand, right hand. Left hand means under inducible system you should write, and right hand means you should write under repressible system. Okay. Point number 2, repressor molecule can bind with the operator. A repressor molecule can bind with the operator so the transcription of the structural genes are switched off. So the transcription of the structural genes are switched off. Right hand. Aporepressor cannot bind with the operator. Aporepressor cannot bind with the operator so the transcription of the structural genes are permanently switched on. Point number 3, left hand. Inducers can bind with the repressor. Inducers can bind with the repressor, rendering it inactive. Right hand. Co-repressor can bind with aporepressor. Co-repressor can bind with aporepressor, rendering it active. left hand. Point number 4. Effector in bracket inducer induces the production of messenger RNA of structural genes. Effector induces the production of messenger RNA of structural genes. Hence, it is called inducible system. Right hand. Effector in bracket co-repressor inhibits the formation of messenger RNA of structural genes. Effector inhibits the formation of messenger RNA of structural genes. Hence, it is called a repressible system. Now, write down how does repressor act. The function of repressor depends upon the effector in bracket inducer and co-repressor. In inducible system, inducer binds with repressor and makes it inactive. Resulting transcription of structural gene to produce messenger RNA. For example, lactose as an inducer binds with Lac repressor and repressor becomes inactive. Thus, synthesis of enzyme occurs. In repressible system, normally the repressor is inactive and addition of co-repressor results active repressor. Active repressor stops transcription and synthesis of enzyme does not occur. For example, tryptophan acts as co-repressor in E. coli. It produces active repressor and stops tryptophan biosynthesis. So, you are given all the probable questions and answers. I think তোমরা ভালো করে পড়বে, সব কিছু দেওয়া হয়ে গেল, এবং এটা ইম্পর্টেন্ট টপিকও বটে। ঠিক আছে, ওকে, বাই।